Hello, everybody. Welcome to the Means of Genes, where we're going to talk about genes and what they means. I'm Ryder Larson, and this is episode 2. And today we're going to be talking about how DNA makes proteins. And previously we talked about what a chromosome is. So we know that chromosomes are in the nucleus of a cell, and we know that they are made up of genes from long sequences of nitrogenous bases like adenine, thymine, guanine, and cytosine. Okay. But what we don't know yet is that every three nitrogenous bases code for an amino acid. Okay, so if we go here, we can see how in this bottom left, you can see this thing kind of got cut off here. I did not intend on that happening, but yes. So every three nitrogenous bases code for a amino acid, and chains of amino acids make proteins. According to Discovery Education, when genes produce proteins, it is called the expression of proteins, and it is the process of coding in the genes being used to construct something in the body, and it leads to traits like eye color, hair color, and height. So our DNA produces these proteins through a process called protein synthesis by using something called messenger RNA. Okay, so RNA, this is going to be a word that you're going to be hearing a lot. RNA stands for ribonucleic acid, and it is like DNA, but it is a single strand. So on the right, you can see this image. It looks like DNA, but it's just like cut in half. So that is what RNA is basically. So it enters the nucleus and then makes not DNA, sorry. It uh, makes DNA split apart, and like it's like, since DNA is like a ladder basically, it's like you can just cut the ladder in half basically. Like that's what RNA does. It unzips it, but it's not cutting it. It's just like unzipping it basically. So uh, RNA enters the nucleus and makes DNA. Oh my gosh, DNE. It's not DNE. It's DNA. Okay. It makes DNA split apart, and then RNA comes in and lines up with half of the split DNA and uses it as a template. And after it, so it goes in, DNA unzips, then the RNA goes in, uses the one strand of DNA as a template. Okay, so RNA comes in and lines up with half of the split DNA and uses it as a template, and they click together to form hydrogen bonds, turning the RNA into a copy of the genetic code. The RNA then slides out, looking like a single strand, like the image on the right, and the DNA clicks together from being separated, and the base pairs connect again. Uh, this first part of protein synthesis is called transcription because the RNA gets transcribed into a copy of the DNA. Discovery Education uses an example of transcription being like writing something on a piece of paper, then on another piece, and then on another piece. They have the same message but aren't the same piece of paper. Same piece of paper. Sorry. um, this applies to RNA because basically the RNA is a copy of the genetic code and it has the same genetic information, but it's basically on a different sheet of paper. That's kind of how that applies to RNA. So RNA also uses a different base other than thymine. It is called uracil. Um, so uracil is used instead of thymine. I'm pretty sure because of like energy, on how like it involves energy, I believe. But uh, but if DNA has thymine, then RNA can use adenine to pair with it because thymine is a base in DNA, but not RNA. And adenine is a base in RNA. So that's how that works. So, anyways, so after the DNA reconnects together, the single strand of RNA needs to cut out the introns in the sequence. So, this is called RNA splicing. Okay. So the reason RNA needs to cut out the introns is because they don't code for proteins. So that like strand of RNA, right? right there, the copy of genetic material does not code for proteins, and we want it to be one continuous code, so the RNA kind of cuts that out, basically. And so after this cuts out, the RNA has to go and deliver the genetic code into the ribosomes in the cytoplasm. And this ribosomes are a whole like different organelle here. So here on the left, you can see nucleus. This is like a little marble-looking thing in here, jelly bean. Ribosomes are here. So this is where the RNA needs to go to use the genetic information and make proteins, because the ribosomes are where proteins are made. So, yes, a whole different organelle, not a nucleus. So, yes, so ribosomes are where the RNA goes to use a transcribed copy of DNA and make it into amino acids. And remember that every three bases or base pairs code for an amino acid, because ribosomes are where they produce the amino acids depending on what the base pairs from the copy of DNA are. Uh, yes, so when every three base pairs code for an amino acid, this is where, in the ribosomes, this is where every three base pairs make an amino acid. So, because the ribosomes are where they produce amino acids. So, yes, the entire sequence of RNA will end up making many amino acids that will form in a chain, ending up with a strand of different amino acids. And these amino acids all together will end up making a protein. And we know this because proteins are made up of long strands of amino acids. And it is important that our DNA makes proteins, because without them, we wouldn't develop parts of our body like muscles, bones, or many other things during human production. And if you're wondering why DNA has to use RNA, it's because DNA cannot leave the nucleus. So let's catch up on slides here. So why can't DNA leave the nucleus? Why does DNA have to use RNA to go send a message to the ribosomes? It's a good question. And DNA is in the nucleus, and proteins are made in the ribosomes. And if DNA ended up leaving the nucleus to go to the ribosomes, it could risk getting damaged. We really don't want that happening, because DNA is really fragile, and this is our genetic information that we're talking about. We just don't want it to get damaged, because it contains all of our precious, genetic information, and damage to it can lead to deformities. And just imagine a strand going through protein synthesis. Imagine what proteins that would end up making. That would not be the best. So, yes, that would be bad. Anyways, this is the conclusion of this episode. We talked about protein synthesis. We talked about how every three base pairs go for an amino acid in the ribosomes. DNA uses RNA to make a copy of the genetic code, leave the nucleus, go to the ribosomes, then end up making a long strand of amino acid. The strand of amino acid end up making proteins. So that is mainly what we talked about today. And thank you for listening in. And in the next video, we're going to go over the topic of inheritance. So thank you very much, everybody. And if you want to see the next episode, then you can go ahead and do so. A second, and then it's recording. Okay, bye.